Hello, my name is Juan. I'm an undergrad student at the Structural Bioinformatics Lab in the National University of Quilmes, and today I'm going to be talking about our work on how chaperones accelerate the evolution on amyloidogenic proteins. To begin with, we're going to introduce amyloids. These are a type of polymer that is mostly conformed by folded beta sheets, that is characterized by being highly packed and highly stable. We can divide amyloids into pathological and functional ones. The pathologicals are proteins that suffer a mutation and then start forming the amyloid, and they are toxic to the cell. The functional, on the other hand, are proteins that can go back and forth into the amyloid state, and they are mostly used for storage. There are a lot of very well-known pathological amyloids, but there is also a big catalog of functional ones. Now, our hypothesis of why they evolve fast comes by mirroring two ideas that are really well established in the field. The first one is that highly expressed proteins evolve slowly. That is because highly expressed proteins must always have the same structure and function to basically function correctly. The second one is that chaperones, when interacting with proteins, can basically let the proteins mutate a little bit. That is because chaperones ensure that proteins fall into the correct state by basically pushing them into the correct structure. This gives a little bit of freedom to the proteins to mutate. Now, I'm going to talk a little bit about evolutionary rates. When we talk about site-specific rates, we're saying that the more important a residue is in a structure and therefore in a function, the less it will change. When we talk about global rates, we are talking about a lot of different factors that can or cannot modulate how a protein changes. We have some of them listed here, like gene expression, length, or IDRs. Now, the dataset that we use is conformed basically by the human reference proteome and all the Homo sapiens amyloid listed in AmyPro. That is the biggest amyloid database in existence. We also gather orthologous proteins from several different species, and we use a single phylogenetic tree for the evolutionary evaluation. We measure the evolutionary rates using CODEM by measuring non-synonymous substitutions between all the proteins in the tree. So what we found basically is that amyloids are proteins that are highly expressed and also that they evolve really fast. This is not how the rest of the proteome work, as we can see here. We can see that in the rest of the proteome, if a protein is highly expressed, it evolves really slow. And we can see that this is according to what is well known in bibliography. Now, what we decided to do is to use a principal component analysis to figure out what global parameters were affecting the amyloids. As you can see here, the biggest component is the first one, and it's mostly composed by expression, interaction with other proteins, and interaction with chaperones. Now, what we did is use a PCR, that is a principal component regression, to see how was the correlation between the evolutionary rates and these components. And we can see here that there is a positive correlation between the first component and evolution. Now, if we look at component number 21, that is mostly composed of only expression, we can see a negative correlation. This is really interesting because it's basically saying that if a protein is highly expressed and it evolves fast, it means that it's interacting with the chaperone to buffer this. On the other hand, if a protein only is highly expressed, it's most likely that it will evolve slow. Now, we can hypothesize that there is a cycle between the chaperone and the amyloid, that basically the amyloid is mutating because it can count on the chaperone to make it fall in the correct way. The problem is that this cycle, this vicious cycle, is broken when the chaperone basically cannot buffer anymore the mutations. And then is when the pathology or the disease for the amyloid is triggered. Thank you very much. I'm running out of time, but I would like to thank the whole team and the Congress. Hope you like the talk.